As we mentioned in our History of Neuroscience lecture, there are two broad fields of neuroscience. There's behavioral neuroscience, which typically does invasive work with non-human animals, and then there's cognitive neuroscience, which typically does non-invasive work with human animals. So we shift now from having talked about invasive techniques such as lesion methods, functional tracing, and single cell recording to talking now about functional neuroimaging. And there are four basic types of tools that we're going to discuss for human brain imaging. There's electroencephalography, which records electrical signals coming from the head. There is magnetoencephalography, which records magnetic signals coming from the head. Functional magnetic resonance imaging, which records blood oxygenation. And then transcranial magnetic stimulation, as well as direct current stimulation, which do not actually record brain activity, they actually manipulate brain activity. They're ways of creating functional lesions. We'll talk about each of these in turn. Beginning with electroencephalography, you can do several types of analysis. Among them are spectral analysis and event-related potentials. Either way, you're talking about voltage differences between pairs of electrodes. Now, electroencephalography in its raw state gives you very noisy, jaggedy, squiggly lines, as we see here. And the trick with electroencephalography is to turn these noisy lines into interpretable signals. If you do spectral analysis, then you're going to come up with one or more various sine waves. These can be as low as 1 hertz in frequency, in which case, if it's 1 to 4 hertz, you're looking at delta. 4 to 7 hertz is theta. 8 to 13 hertz is alpha. 14 to 30 hertz is beta. And around 40 hertz is considered gamma. These are just Greek letters given to each of these frequency bands. And each frequency band tends to reflect specific psychological processes. So for instance, if you're interested in attention, you're well off focusing on alpha in the 8 to 13 hertz range. If on the other hand, you're looking at memory, you might want to focus on theta in the 4 to 7 hertz. If you're interested in consciousness, then you might want to focus on the gamma range up around 40 hertz. Either way, you're talking about taking that noisy squiggly line and converting it into a finite set of sine waves. In fact, what you're really doing is doing a fast Fourier transform on that noisy line in order to get clean sine waves out. And as I said, different sine waves tend to relate to different psychological processes and are useful to that extent. These signals come from not the axon, but the dendrites in the form of excitatory and inhibitory postsynaptic potentials, as we talked about before. So, alternatively, instead of using spectral analysis, you could use event-related potentials. Either way, here's a look at the cap. No matter what kind of analysis you're doing, you're going to use this kind of EEG cap. In this case, we see a cap that has about 64 electrodes in it. And you can get these squiggly lines out, or you can make nice pretty pictures by showing kind of hot spots, basically positive voltage in red and negative voltage in blue. Event-related potentials look different than the sine waves we get in spectral analysis. In event-related potentials, we get sine waves, and you can measure these. Well, I'm not sure I said that right, but they're called waveforms. You can measure these waveforms for, say, old and new items. This is from an actual memory experiment. But you can use event-related potentials for any psychological process you'd like to study. Now let's talk about the relative resolution of electroencephalography. EEG has excellent temporal resolution. EEG can measure brain activity as fast as that brain activity is occurring. And so there's really no reason to wish for any greater speed in terms of your temporal resolution. Spatial resolution, on the other hand, is rather difficult. It's poor. Knowing when a signal is happening is very easy with EEG. Knowing where in the brain it's happening can be very, very difficult. So skipping here, let's talk about why it is difficult to know where in the brain the signal is coming from. And there's a couple of reasons for that. One is conductivity. If we look over here, here's a slice of brain, and we see electrical currents going through the neurons, through the brain. Those currents have to propagate through the brain, through the meninges, which include the pia mater, the arachnoid space, and the dura mater. And then they have to get through some spinal fluid. Then the electrical signal has to get through the skull, which is filled with billions of air vesicles. And then it has to get through the scalp before that signal makes it to the electrode. And that means there's lots of opportunity for distortion of the signal. Each one of these layers here is not a particularly good conductor of electricity. Therefore, each time it goes through one of these layers, the signal gets smeared out. And so we say that EEG has conductivity problems. But it also has an even more fundamental problem known as the inverse problem. The inverse problem is stated basically for any pattern of electrical activity you get in your electrode, there's an infinite number of ways the brain could have given you that signal. An infinite number of ways the brain could have given you that signal. You're looking for the one true way that the brain gave you the signal, but in order to know what that one true way is, you have to eliminate all the other possibilities. But there's an infinite number of those possibilities. Therefore, you can't eliminate them. Therefore, you can't ever really know truly which one of those solutions is actually the one that the brain used. So, to restate, the two issues with this that give rise to poor spatial resolution in EEG are conductivity and the inverse problem. Now let's look at a similar technique called MEG, or magnetoencephalography. And it really is very similar to EEG because it's effectively the same signal in many respects. That is, anytime you get an electrical current, there's a magnetic field associated with that. Therefore, any electrical current going through the brain is generating a magnetic field. And instead of measuring that electrical signal, you can measure the magnetic signal instead. It gives us squiggly lines very similar to what we see with EEG. And again, you can do spectral analysis, or you can do not event-related potentials, because potential refers to electricity. In MEG, it's called an event-related field to reflect that it's magnetism. Like EEG, MEG has excellent temporal resolution, and we might argue that it has moderate spatial resolution, because even though MEG still has that really difficult inverse problem that EEG has, it does not have the conductivity issues that EEG has. The scalp, the skull, the meninges, etc., they don't really interact with the magnetic field, so they don't smear it out, which means that MEG only has one of those problems, the inverse problem, not the conductivity problem. But otherwise, it's very similar. However, it's much more expensive. We can see over here that we can see this dewar, as it's called, and in the bottom of this dewar are the magnetometers, the sensors which are recording the magnetic field. But these magnetic fields coming out of the head are extremely small, and in order to detect these magnetic fields, you have to supercool the magnetometers. So this dewar has liquid helium cycling through it in order to supercool the magnetometers. And as such, these magnetometers are called SQUIDs, superconducting quantum interference devices. And what cost so much money is not so much the doer, which is a little more expensive than EEG, but not super expensive. What is particularly expensive for MEG is the need for the magnetically shielded room. Now let's talk about magnetic resonance imaging. And the first thing to note here is that this has the word magnetism in it, much like the term magnetoencephalography does. However, the sources of magnetism in these two techniques are very different from one another. As we said, with magnetoencephalography, it's your brain's electrical currents that are generating the magnetic field. So your brain is generating the magnetic field that's recorded in magnetoencephalography. In magnetic resonance imaging, the researcher is creating a magnetic field and then placing the body inside that magnetic field to see how it is distorted. So one can get down into the nitty-gritty of the physics of all this. We won't bother to get too deep, but we do want to know that effectively what's going on is when you place a body inside of the magnetic field, the protons in the atoms inside your body all line up with that magnetic field. And then you deliver a radio frequency pulse to the tissue, which knocks the precession of those protons out of alignment with the magnetic field. But they will slowly, well, I say slowly, but it's actually extremely fast, but they will return back to the alignment with the magnetic field, and different material aligns quicker or slower. So for instance, information or protons in white matter will realign very quickly, whereas protons in gray matter will take longer to realign. As such, you can break this space in the magnetic field down into not pixels like we get on a screen, but voxels, because these are three-dimensional. You can measure the magnetic field at each one of these voxels. And that works something like this. The magnetic field space is, you measure at each one of these little 1 cubic millimeter voxels, and you can get a picture of the brain out of there. But that's magnetic resonance imaging, or structural magnetic resonance imaging. Like we see on the upper right image here, that is a structural image. It's just giving you a picture of the structure of the brain. What psychologists are typically interested in is fMRI, functional magnetic resonance imaging. In this case, instead of measuring the tissues in the brain, we're measuring blood flow or blood oxygenation. And this gives rise to the bold signal, the blood oxygen level dependent signal, the bold signal. And of course, whatever part of the brain you might happen to be using, that area of the brain will be depleted of energy and need a resupply of oxygenated blood. And you can see this flow of this blood. And so to go with our memory example in the hippocampus, we can say that the hippocampus, if you use it to retrieve a memory, you're going to deplete the energy in the hippocampus, and therefore you get a rush of blood to replenish that. Functional MRI has excellent spatial resolutions. It's the best technique out there if you want to see exactly where the brain activity is coming from. However, the temporal resolution of functional MRI is actually quite poor. It's very difficult to see exactly when brain activity is happening. And this is due to the sluggish nature of the hemodynamic response, or the sluggish nature of the bold response. It takes blood several seconds to travel to the brain, yet the brain activity is happening on the order of milliseconds. So the temporal resolution is quite poor, even though the spatial resolution is quite good. Now, how do we use this stuff in the mind? Here's an example of a memory experiment in which participants are lying in a scanner and they're going to see a list of study items, and they're told to study those. Now, in this particular experiment, the researchers were not interested in how does the brain encode the information during the study phase. They're interested in how does the brain retrieve the information later in the test phase. So during the study phase, the scanner was not turned on, but participants lay there and looked at various like a firefighter, and were just saying for each item they saw, is it living or non-living? So in the case of firefighter, obviously the answer would be of ladybug, calculator, mountain, non-living. In reality, there were about 150 of these items that they had to study. And then there was a short break and the scanner was turned on. Now that the scanner's on, be given items to say whether they're old or new. So anteater in this example would be new because you didn't see it in the phase. Ladybug would be old, calculator and boat would be new. And there'd be about 100 of these items. Half of them old, half of them new. Now what? Well, we need to do subtraction. Because what we want is this image here, the retrieval image, the brain. Where are those voxels in the brain that were significantly active during retrieval? However, as you could see, the task not only involved memory retrieval, but it also involved vision. You had to see the words, and it involved language. You had to read the words. And so when we look at our old image, or that is our item, our functional MRI image for when participants saw old items, we see a lot of activity, but not all of that is going to be memory-related activity. Some of it's going to be vision, some of it's going to be language. So what we do is take the brain image from new items that have all the same vision and all the same language, but lack the retrieval-related activity. We subtract those voxels from these voxels, and the image we're left with is retrieval-related voxels specifically. No vision voxels are in here, no language voxels are in here, just retrieval. And by the way, if you look at this retrieval image and say it doesn't appear to have come from the old minus new images, well, you're somewhat correct. This retrieval image has had some other stuff done to it. So it's not exactly the subtraction of old from new, but the general idea is the same. Okay. And what functional MRI gives you is not pictures of blood flow, it gives you pictures of statistical probabilities. So as we look over here, this red and orange and yellow represents very high statistical significance. Red is a little lower significance. But each one of these voxels is active. It's activated. It tells you there's a high statistical probability that blood flow changes happened here during your task. And finally, we have the two techniques which are not actually brain imaging techniques because they don't make images of the brain. They are ways of manipulating brain activity. You can either excite a brain region, causing it to fire more action potentials, or you can inhibit a brain region, causing it to fire fewer action potentials. So we will kind of talk about these together: transcranial magnetic stimulation and direct current stimulation. This is not too different than what you heard about in the old days with electroconvulsive therapy, only it's much lower doses and being much more responsibly used here, and it doesn't do any actual harm to participants. So these are ways of creating functional lesions. We say functional because they're not actually lesions where you're damaging brain tissue. You're just impairing the function of a brain region temporarily. They're also called transient lesions because they don't last long. Basically, you can turn on TMS or direct current stimulation and cause a brain region to hyperpolarize so that it won't fire action potentials. And then you can get a participant to perform a task. So to continue with our memory experiment, even though it's unlikely that TMS or DCS can reach the hippocampus, we'll go ahead and use it for our example here. So imagine you use TMS to inhibit the hippocampus. Well, the person will have trouble retrieving memories during that time. And as soon as you turn the magnet off, the hippocampus goes right back to its normal functioning. So it doesn't really create any actual damage, just transient functional impairments. And the way this works is with TMS, you're using a magnetic field. If you look at the image here in the top right, we see an MRI image that was taken of a participant, and then the magnetic coil for the TMS was applied to a particular brain region. So again, TMS doesn't give you this picture. This picture is coming from MRI. TMS just allows you to manipulate the brain activity. And it's very similar with DCS. It's going to, instead of passing a magnetic field through the brain tissue in order to hyperpolarize it, DCS, direct current stimulation, is going to pass an electrical current through the brain tissue in order to hyperpolarize it. Now that's for research purposes, you're looking to hyperpolarize tissue, but oftentimes in clinical settings, you're trying to excite a brain region. So for instance, it's fairly common to use TMS or DCS to stimulate parts of the brain to help alleviate depression. I personally am not well read on this literature, so I don't know just how efficacious these techniques are, but I do know they're fairly popular. So again, you can inhibit or hyperpolarize brain tissue to measure the effects of a transient lesion, or you can stimulate or depolarize neural tissue in order to help with various clinical issues such as depression.